Ok, buenas tardes nuevamente a todos quienes nos están acompañando, comunidad del CINCCO y público en general que asisten a este seminario transdisciplinar del Centro de Investigación en Ciencias Cognitivas de la UAM. El día de hoy nos acompaña la doctora Chiara Mazzi. Ella hará su presentación en inglés. Entonces nada más voy a dar algunos puntos importantes que tenemos que considerar. Les vamos a pedir que mantengan sus micrófonos y cámaras apagadas durante la presentación para que no haya interrupciones. La doctora Chiara Mazzi me pidió que si hay preguntas que sean fundamentales durante la presentación para comprender algún concepto, sobre todo si consideran que es demasiado técnico o no fue muy clara, se lo pueden preguntar al momento, de tal manera que lo pueden escribir en el chat y yo haré la comunicación con ella para aclarar el punto. Si la pregunta no es fundamental realizarla durante la presentación y pueden esperar hasta el final de la misma, pues les agradeceremos esperar y al final daremos espacio para preguntas y respuestas más generales y de curiosidades que puedan haber surgido con el tema. Las preguntas las pueden escribir en inglés para la doctora o en español. Yo puedo proceder a traducir la pregunta en la medida de lo posible. Y bueno, si hay alguien aquí que también habla italiano, puede escribirla en italiano. También de todas formas tendré que traducirla al inglés para que todos se puedan enterar de qué iba la pregunta. Y bueno, sin más, entonces voy a pasar a a presentar a la doctora en inglés. Okay. Good afternoon, everyone. Welcome to our seminar on transdisciplinarity. As you know, this seminar aims to share scientific knowledge in topics involving diverse disciplines in cognitive science. Today, I'm really glad to introduce our special guest, Dr. Chiara Mazzi, with the talk investigating perceptual awareness by means of Phosphenes. In brief, Dr. Chiara Mazzi is fellow researcher in the Department of Neuroscience, Biomedicine, and Movement Science at the University of Verona in Italy. Dr. Mazzi's research interests focus on the investigating of the visual brain and the spatiotemporal dynamics underlying the subjective conscious experience of seeing. Her research aims to understand how perceptual awareness is generated both in the intact and lesioned human brain by using a combination of non-invasive neuroimaging techniques such as TMS, EEG, and NIRS. She has a long record of participation in scientific events and paper publications. So I want to say welcome to Dr. Mazzi in our Centro de Investigación en Ciencias Cognitivas. Welcome, and you can start with your presentation. Thank you so much.
Okay, thanks, Javi, for your nice presentation and for the invitation. As you have already told, today I'm going to talk about perceptual awareness and in particular visual awareness, which is the subjective conscious experience of seeing occurring in our everyday life whenever we see an object like an apple, for example. So we know that when our retinal cells transduce light into electrical signals, triggering thus a cascade of neural events along the visual pathway that lead to a conscious percept of the specific object we are looking at. However, despite the increasing efforts over the years and decades, the nature of conscious awareness remains a mystery, and many questions still remain unanswered, such as how, when, and where the visual experience is actually generated in our brain. So my goal is to highlight the so-called neural correlates of perceptual awareness, which are defined as the minimal set of neuronal mechanisms necessary and sufficient to give rise to a conscious percept. Of course, many theories and models about this topic have been proposed over the years. One of the most controversial issues concerns the role of the primary visual cortex about its role. Okay, so according to an influential model, the Lammes model, feedback to primary visual cortex after a first feed-forward sweep has a causal role in the emergence of visual awareness, suggesting thus that the integrity of V1 of the primary visual cortex is necessary for consciousness to emerge. So the idea that V1 is directly responsible for conscious vision originally comes from the fact that a lesion to this area leads to blindness in the corresponding portion of the visual field. So as you can see here on the left, you have a representation of the intact visual field characterized by a normal vision. Here on the right, instead, you can see how lesion of the the right primary visual cortex results in a complete loss of vision in the contralateral visual hemifield. According to another influential model proposed by Milner and Goodale, visual processing is characterized by two different streams. The ventral stream originating in V1 and projecting to the inferior temporal cortex, which is perceptual in nature, and then we have the dorsal stream projected to the superior parietal cortex. In this model, the visual awareness is restricted to the ventral stream, while the dorsal stream serves to convert visual input into actions, and thus is usually associated with the processing of the position and motion of the presented stimuli, concluding that visual product of dorsal stream are not available to conscious awareness. So basing on these two models, it seems that neural correlates of consciousness can be found along the ventral stream only, and then that the integrity of V1, so the primary visual cortex, is needed for consciousness to emerge. So to test these two statements, we designed a series of TMS experiments employing Phosphenes to try to answer different experimental questions that we will see in a few minutes. So let me first define what a phosphene is and why we decided to use Phosphenes. I try to represent a phosphene in a graphic way. So you will see something, a lightning appearing here, which stands for the single pulse TMS. And then after that, you will see something very brief appearing in this portion of the screen, which represent, of course, the phosphene. Basically, a phosphene, it's a light sensation in the absence of any external light source and in the absence of any other visual input from the outer world. Usually are evoked by single pulse TMS over a cortical visually responsive area. We can say that Phosphenes are a faint grayish percept, which are a few visual degrees in size and are located contralaterally to the stimulated cortical sites. The position of this percept depends on gaze direction, meaning that shifting the fixation cross, like here, for example, the position of the percept will thus shift accordingly. The visibility of this kind of percept is possible both with open and closed eyes. Then here on the right, you can see some graphic representation reported by subject during the experiment. And as you can see, the position, the shape, and also the size of phosphine can vary a lot from trial to trial, but also among the different subjects tested. So most of the time we have phosphine similar to this one, but sometimes we can also have, we can say, a changement in the texture of the screen you're looking at during the experiment. In particular, we chose to use phosphine for different reasons. First, because they can be used to explore the neural dynamics underlying visual perception, though in absence of any external stimuli. Then, because their processing has been shown to resemble that of real external stimuli, so they are reacted to faster as their brightness increases, and they show an interhemispheric transfer time in line with that of healthy subject in response to real external visual stimuli. And finally, we chose to use phosphine because at the same time, they allow to assess the visual cortical excitability by identifying the minimal intensity needed to evoke a percept. But let's start from the first experiment, whose aim is to understand if the dorsal stream can be sufficiently effective to generate visual conscious impression by itself, so independently from the contribution of V1. We will test both healthy participant and a myopic patient using, of course, TMS and using psychophysics measure. So previous evidence has shown that phosphines can be elicited also stimulating the intraparietal sulcus along the dorsal stream, where additional retinotopical maps have been recently reported, though being less precise and less organized than those of the occipital cortex. So the fourth experiment aimed at determining whether these phosphine elicited stimulating IPS could be generated with a contribution from the occipital cortex or not. To this aim, we tested first a group of healthy participants characterized as by a normal brain architecture, and we stimulated V1 and IPS in different experimental sessions, and we contrasted then the corresponding phosphine threshold and other perceptual features such as brightness, vividness, the eccentricity, and area. Our reasoning was that if IPS is an independent generator of conscious visual percept, we should find reliable differences in these parameters. So here, the psychometric function for the two sites of stimulation are presented. In particular, we have in blue the psychometric function for occipital phosphine, and in red the psychometric function for parietal phosphines. And you can see that the threshold, the 50% threshold for occipital cortex is much lower than that of parietal phosphines. Moreover, when healthy participants were requested to draw and rate their percept, as represented here, where on the top we have occipital drawing, phosphine drawing, in the bottom part of the slide, we have parietal phosphene drawings. On the left, we have stimulation at phosphene threshold, while on the right, we have the stimulation at suprathreshold intensity. So parietal phosphines in general resulted to be significantly less vivid, less bright, more variable in size, and the eccentricity of parietal phosphine was not modulated by the stimulation intensity. And this could be due to a coarser, less precise retinotopic organization of IPS than V1. Instead, stimulating the occipital lobe, the higher the stimulation, the more the phosphine were lateralized. Moreover, as already said, we found that a higher TMS intensity was required to perceive parietal phosphine with respect to that needed to elicit occipital phosphine. This finding is in line with previous fMRI findings, indicating that given the same amount of visual stimulation, the bold activity represented here on the y-axis, so the bold activity in IPS, represented here, is much smaller than the bold activity registered in the primary visual cortex, represented here. So the response amplitude decreased, gradually moving into higher parietal areas. So to corroborate these findings obtained with healthy participants, we induced parietal phosphines into 2 immunopic patients with a lesion in their primary visual cortex. Just few information about these 2 patients, patient SL and patient AG. So immunopic patients are affected by definition, by visual field effects due to cortical damage, in this case, as a result of an ischemic stroke. Their visual loss is homonymous, meaning that the same region is affected in both eyes, as highlighted in black in both the perimetries, sorry. And the structural MRI, represented here, evidence a lesion involving their primary visual cortex, as you can see highlighted in red. In addition, a full field visual stimulation using fMRI did not show any activity in correspondence with their lesion. Finally, we tried to stimulate at suprathreshold intensity over their lesion, and this stimulation did not result in any conscious visual perception, so no phosphines at all for them stimulating their lesion. And this exclude again the presence of residual activity within their V1. So all this information together confirm a complete and circumscribed V1 lesion, making thus this patient critical to test those models, postulating that V1 and feedback to it are necessary for awareness to emerge. So here we have the results on the left. So on the top part of the slide, we have results for patient SL, while on the bottom we have results obtained with patient AG. So on the left, you can see the stimulation sites, which correspond both to their ipsilesional IPS. So this is their lesion, and this is the stimulation site. So this is IPS. And the same thing, but on the other side, since the lesion of patient AG was on the right, so this was the stimulation site along his right IPS. In the middle column, there are the obtained psychometric functions, where again we have the intensity stimulation, the stimulation intensity over the y-axis, and the percentage of perceived phosphine in the y-axis. So the fact that we have these two psychometric functions means that both patients could reliably perceive and report phosphines following single pulse TMS over their ipsilegional IPS. Importantly, their parietal phosphine threshold was comparable to that obtained stimulating the parietal cortex in healthy participants. On the right, there are patient phosphine drawings, meaning that they could position them within their blind hemifield, which was not obvious. And again, as happened with healthy subjects, their phosphine increased in size as TMS intensity increased. While again, the eccentricity was not modulated by the TMS intensity, but their phosphines remain at the same spot, also increase the intensity. Finally, both patients was able to score their percept either in terms of vividness and brightness, and those scores were similar to those obtained with healthy subjects. So looking at the result of this first experiment, whose aim was to assess if the dorsal stream can generate visual awareness, we can say that the stimulation of IPS results in conscious percept, and importantly, this is true also for hemianopic patients with a complete V1 lesion. So taken together, these results advocate for a distinction between the two stimulated sites in terms of the locus of generation of phosphine, meaning that there is initial evidence suggesting that dorsal stream could be considered an independent gatekeeper of awareness, at least under these circumstances. And V1 seems not to be necessary for awareness to emerge. Let's move to the second experiment, which instead aimed at investigating the spatiotemporal dynamics of feedback from IPS to the occipital areas, given that the possibility that an involvement of the contralateral hemisphere in parietal phosphine generation cannot be totally excluded, especially in hemianopic patients. So in this case, we use the event-related optical signals called EROS, which is a neuroimaging technique using infrared light through optical fibers like these ones to measure changes in optical properties of cortical brain areas. In particular, this technique detects variation of the scattering properties of the neuron themselves, providing thus a good temporal resolution. So the main strengths of this technique is a good temporal and spatial resolution, while the limitations are represented by a low signal-to-noise ratio and a reduced penetration of the light into the head, which makes it difficult to reach brain structure distant from the scalp or surrounded by white matter. So going back to our experiment, for sure we know that parietal and occipital cortex are highly interconnected, and for this reason we stimulated the left IPS with single pulse TMS, and simultaneously we recorded fast optical imaging to measure the resultant interregional propagation of TMS-induced activity. So fast optical imaging of bilateral occipital cortex and right parietal cortex revealed only a transient activation of left occipital cortex, so this ROI, in the time window ranging from 24 to 48 milliseconds following the TMS of left IPS, thus demonstrating a clear effect of parietal stimulation on the ipsilateral visual cortex activity, while no activity was recorded on the contralateral hemisphere. So looking at these results, aiming at investigating a possible involvement of the contralateral hemisphere in the generation of parietal phosphenes, we can say that the stimulation of IPS confirms a strong relationship between parietal cortex and the ipsilegional occipital cortex, while failed to find significant modulation in the contralateral hemisphere, supporting thus the independence from the contralateral hemisphere of parietal phosphene obtained in immunopic patients. In the third experiment, we try to better characterize the neural dynamics underlying phosphene perception in occipital and parietal cortex by combining TMS and EEG recording. And again, we are going to test both healthy participants and immunopic patients. So this combination provided the possibility to modulate with TMS the brain activity, the ongoing brain activity, and at the same time to measure with EEG the induced cortical activation with a millisecond time scale, which characterize EEG. So a relevant advantage of this combination is that the TMS bypasses peripheral and subcortical structures, so that TMS evoked potential do not depend on the integrity of the sensory system. In particular, I've compared TMS evoked potential where the phosphene was present with TMS evoked potential where the phosphene was absent. In this way, the subtraction of phosphene absent trial from phosphene present trial can isolate the differential activity related to conscious phosphene perception and removing the non-specific responses to the typical acoustic or somatosensory artifact, which are identical across the two conditions. So here on the left, I represented TAPS elicited by TMS pulses over V1, so occipital cortex, where phosphene present condition is represented in red, and phosphene absence condition is represented in black. So the two conditions differed significantly in three late time windows highlighted in gray, occurring after 300 milliseconds. Looking instead at this graph, where time is represented on the x-axis, so 0 from 600 milliseconds, and electrodes from occipital lobe to frontal lobe are represented on the y-axis. The first significant cluster of electrodes showing differences between phosphene present condition and phosphene absent condition. The first cluster was found in temporal electrodes in an early time window at around 80 milliseconds after the stimulation of occipital cortex. These are the result obtained stimulating IPS, so parietal cortex, which is along the dorsal stream. In this case, underneath the stimulation, so under considering the electrode PZ, we observe also an early critical time window at around 80 milliseconds following the stimulation of the intraparietal sulcus, which was followed by late activity around 300 milliseconds, as already found for occipital stimulation. Looking instead at this graph on the right, the first significant cluster was found at around 100 milliseconds, and in this case, significant electrodes were located in centroparietal electrodes. So this map represents scalp current density map, SCD, and display the possible generators of the effect for the early time window. So here reflects an parietal activity, while earlier the first cluster was reflected by an activity in the temporal cortex. Again, we tested patient SL, and again we tested her ipsilegional parietal cortex, and her TEP highlighted the same critical time window seen in healthy participant, which was not followed by a massive later activity in a later time window. So given that temporal sites did not show any effects, it can be concluded that these sites, so the temporal sites, are not part of the functional network generating parietal phosphine, thus reinforcing the idea that parietal cortex can be an early and independent generator of phosphenes. So taken together, the data of the present experiment show that the time course of phosphene perception following occipital and parietal TMS is different, so a different time course for the two sites of stimulation, with an earlier onset for parietal than for occipital phosphenes. In particular, different area seems to be responsible for the emergence of awareness. Temporal areas were critical for occipital phosphenes, while parietal areas were critical for parietal phosphenes. In the fourth experiment, we tested a patient whose lesion was bilateral, offering the opportunity to investigate whether hemispheric differences exist in the generation of perceptual awareness. As already said, this patient shows the so-called altitudinal hemianopia in his upper visual field due to a bilateral damage to the lower bank of his primary visual cortex. So differently from the previously tested patient who were characterized by a complete V1 lesion, and thus no phosphine could be elicited by stimulating the occipital cortex. In this case, his lesion offered the possibility to induce phosphine by stimulating both occipital and parietal cortices in both the hemispheres. So in this case, for this patient, we had 4 different stimulation sites. So patient AM could experience phosphines described as brief, static, and mostly grayish. When TMS was applied over both occipital and parietal cortices in both the hemisphere. So in all the 4 stimulation sites, the patient reported to perceive the phosphine in his upper and blind visual field, and this is a little bit weird. As a novel evidence, moreover, patient AM could also reliably report phosphine while his occipital cortex was stimulated, so differently from the patient we have seen before. And finally, the threshold for phosphine detection was higher for parietal than for occipital TMS, again in line with previous findings. So importantly, for both the occipital and parietal, so occipital and parietal stimulation, in both hemispheres, it was possible to find a cluster of significant electrodes in posterior sides ipsilaterally to the stimulated hemisphere, showing differential activity for presence versus absence of the perception of phosphines. And this differential activity peaked for all the stimulated side at an early time window, which is around 70 milliseconds after TMS, so in line with what we have seen before. So the present data and those already present in literature corroborate the evidence in ERP studies in favor of early correlates of visual awareness, and suggest also that posterior brain areas can serve as early gatekeepers of perceptual awareness in the human brain. So trying to sum it up, results obtained reveal and confirm that if on one side the primary visual cortex is certainly important for normal conscious vision because it clearly creates the condition to perceive the outer world, on the other side, V1 is not strictly necessary because perceptual awareness can emerge even in its absence, supporting the growing body of evidence stating that the primary visual cortex is not causally involved in eliciting conscious perception. So indeed, our results demonstrate that the dorsal stream, which is traditionally thought to be unconscious, could instead generate visual awareness, at least under these very basic circumstances. Finally, these data show that phosphine can be considered an ideal tool to directly study neural correlates of consciousness both in healthy and lesion brain. So thanks for your attention and thanks to my collaborator.
Thank you so much. It was really interesting, at least for me. If you have any question, you can write your questions in the chat, or you can ask for to open the microphone for the question. Si tienen dudas, pueden hacerlo por el chat o pueden pedir que se abra el micrófono. Yes, we have a question for the CINCCO direction. You can open the microphone, I guess.
Yes, Dr. Mazzi, thank you very much. Very exciting data and experiments that you carry on. I have like too many questions, so that I would like a private session. But I just mentioned 3 or 4, and you can react of whatever you want. With the conclusions, I may say that it's a way. I mean, one of your conclusions, I think, point at the possibility of explaining blindsight, right? Because usually subjects don't report an experience, but are able to move in space successfully. So that's one thing. Another is that you don't mention color experience in the Phosphenes. I would like to know whether they are supposed to be color or if they report any color for the Phosphene impressions. Then I wonder if you could also find similar results with dreams, which is a spontaneous activity and probably more complex than Phosphenes, and if that could corroborate, your findings. And finally, as a philosopher, I would object to the idea that people are perceiving anything. I don't think they perceive anything, because perception is supposed to entail existence of the object perceived. So this is just a endogenous brain activity that correlates with some subjective experience, which I would call visual impression, but not perception, because of a conceptual argument that...
So are you referring to Phosphene, right?
I'm sorry?
Are you referring to Phosphene? So you are saying that Phosphene is not perception, but sensation?
Yeah, or I mean, like visual impression if you want, but it doesn't entail perception insofar as the object does not exist. So when you're saying that Phosphenes is a way to study perception because visual stimuli are somehow similar, I would say that no, there is a radical difference between visual stimuli coming from the real world and an endogenous activity. The first one is a feed-forward signal that also has a feedback return, in the signal, and uses the retina. And these Phosphenes are just activity of the occipital brain areas. So I'm sorry for the length, but if you want to react to whatever of these questions, I'll be happy. Thank you very much.
Okay, thanks for your questions. Starting from the last one, I understand when you say that Phosphene are not similar, are not perception, but try to see Phosphene as the simplest form of perception, okay? It's true that Phosphene are really basic phenomenon, so they don't, they are not characterized by a rich experience as what we can perceive in the outer world. And also with Phosphene, we have one object at a time, while in the real life, in everyday life, we are into a very rich context where potentially everything can be perceived. But for sure, it's something that can be reported also by a myopic patient, and they can report something in their blind field, in blind hemifield. So it's the very basic form. I would say it's a very basic form of perception, but I understand your point of view. Then I remember something about the color experience. Just usually phosphines are reported to be grayish or white, but it can happen also that some participants report reddish, greenish, and yellowish perceptions, and this is often the case when people are looking at colors before the stimulation of their cortex. And what about, okay.
The question about the blind side is something interesting for me. What does Phosphenes say about blind sight?
Okay. So blind sight. So what was the question? Okay, I think that the question was about my conclusion regarding blind sight. So this patient didn't show blind sight, which is a good performance in absence of perception. Okay, in this case, patient could report in a reliable way what they perceive. So in this case, we are talking about perception, not any absence of conscious report. So my conclusion are not about blind sight, but on the opposite side. So my data are about patient reporting something. So they are totally aware of what they see, of what they perceive. Did I answer, Javi?
Yes. I have a couple of questions about the contributions of IPS and superior parietal areas in this, in both consciousness and blind sight. But I will leave my questions at the end of this section of questions and answers, because there is another question from Janine Rodiles.
Thank you, doctor, for sharing your very interesting studies. I would like to question about from your results. Would you say that an stimulation in the occipital and parietal area would provoke a better function of the visual cortex? This is my first question. I'm not obviously expert in this area, but I wondered about the schizophrenic patients where they don't have apparently damage in the visual cortex, but they have during the hallucinations, especially visual hallucinations, they have completely changed perception. Could you say something about this from the studies that you present today?
Okay. So I wouldn't say that stimulating their occipital and parietal cortex, their visual functionality would increase, because the idea is that with TMS we are like injecting some kind of noise in the ongoing electrical activity. And in this case, we produce something, so the perception of a phosphine. But I'm not sure this can be used to improve the functionality of cortex. Probably stimulating visual cortex of this patient, we will affect their visual functionality, but I would not say that we will improve their functionality.
Okay, and the second was about the schizophrenic patients.
There was another question. Sorry, I couldn't remember the second question.
Yeah, the second question. Can you hear me?
Yeah.
Okay, the second question was about the schizophrenic patients and the hallucinations. I remember that before this presentation, I asked you about the hallucinations. I tell you, maybe phosphenes are something similar to hallucinations, and you say, if I don't remember, but you say no. Phosphenes are not hallucinations. So, yeah.
So, I'm not an expert in visual hallucination, but and I don't know where this hallucination come from. Okay, so I don't know the origin of these. Are they visual hallucination or auditive, auditory hallucination? Usually.
Yeah, in patients, in schizophrenic patients, they have both, I guess.
Okay, but did you mean to use phosphine as a rehabilitation, or so? What's the aim of inducing phosphine in these patients? So that's my question. Sorry.
I'm not sure. Janine, can you repeat your question about the schizophrenic patients? Janine, are you there? ¿Estás allí, Janine? Okay, I think that the question regards maybe the brain contributions to the hallucinations, not the use of phosphenes for the rehabilitation. I think that the question was related to if the same areas producing the hallucinations in schizophrenic patients are the same areas that can produce the phosphenes.
Probably, yes. I mean, the visual brain is really spread along the human brain, but we can hypothesize that the areas generating visual hallucination and area potentially generated phosphine could overlap. But I'm just guessing. I mean, I don't have a real answer.
Okay, yes, I know that you are not working with psychiatric patients. So if you have another question, you can put your hand up, or you can write it in the chat. In the meantime, I wanna continue with the question about the contributions of IPS and superior parietal in the phosphenes. Because in the phosphenes and also in the blind side, maybe I know that maybe blind side is not your field of interest, but I remember in the last work that we found the contributions of a superior parietal cortex in the presence of blind side patients. That it was not found actually in patients with lesion in this area. So you are stimulating the inferior parietal sulcus. Have you tried to stimulate other areas?
Okay, potentially. I think that I haven't tried yet, but I think that potentially we can get phosphenes stimulating a lot of different areas. So so far we know phosphine stimulating V1, V2, and V3. Okay, and then we have moving phosphine stimulating MTV5, and then now we have phosphine stimulating parietal cortex. But in the past, CZ, so the highest point of the head, was used as a control site, but also in that case it was possible to elicit visual sensation. So potentially we can stimulate any visual responsive area, eliciting some phosphines. Then a different question, which is more difficult to answer, is whether the origin of this phosphine is every time different, or the origin can be reduced to some critical areas. And I don't have any answer about this.
Yes, I think that is a really interesting field, but there are a lot of questions that we don't have answer yet.
Yes,
so,
a lot.
Yeah.
But just a few words about blindsight again. It's difficult to report blindsight with phosphine, since by definition in blindsight patient they are not reporting, they are not actively perceiving something, but their behavior is above chance, because since with phosphine I'm not presenting anything on the screen, and since they do not report anything, how can I infer that something were there? Okay, of course we can use indirect way to measure the presence of something, like reaction times with force choice task, or I don't know, the measure of the pupil. But I mean, the reportability of phosphine is the key concept. With blindsight, by definition, patients are not aware of what they are seeing.
Yes, it makes sense. No, maybe we can try to produce the phosphine at the same time that you are presenting a visual stimuli. Mm-hmm. I don't know if it is possible.
I don't know, but we can think something about it.
Dirección CINCCO, do you have another question?
Yeah, it's just to pursue the matter. I think it's a very interesting issue here. Dr. Mazzi, you concluded that the dorsal stream seems to be sufficient to evoke awareness of phosphenes, and we know that the dorsal stream is also related to action and motor activity. So going back to the blindsight case, I wonder if it is a way to somehow reinforce the idea that blindsight patients do actually perceive, are aware of something, perhaps below threshold of awareness, but are able to extract information from the environment.
They are able to, sorry, I couldn't hear.
They're able to extract information from the environment,
mm-hmm.
Although it might be below threshold. But what I'm saying is that the dorsal stream is working because it's related to movement, and it's also demonstrated from your work that awareness is possible. There's some kind of awareness. In the case of blindsight, they don't report experience, the patients, but it might be just because it's below a threshold, and that they don't report it, but they're somehow able still to detect to extract information from the environment and have a successful behavior.
I agree with what we are saying. Then it depends on what you think blind sight is. I mean, there is a debate about the nature of blind sight. Is perception under a threshold, or is, I mean, it depends also on how do you define blind sight behavior. But I agree with your interpretation.
Yes, there is a discussion if blind sight patients can see something or not, or is only a motor response without consciousness. No? But there are different points of view in the literature and in the different groups studying blind sight. I remember that.
And you
I'm
know
still working. Okay, we have another question from Jaime. Are you there, Jaime? Jaime?
He's gone.
I don't know. Jaime, are you there?
¿Sí me escuchan?
Yes.
Okay. Okay. I was saying, first of all, thank you very much for your presentation. Congratulations. And my question is more about the methodological stuff. I'm not an expert, but I would like to know about what's the use or what's the advantage of using both TMS and EEG together? Well, I know the limitations of EEG. But there might be special limitations, but how can TMS complement your study for a better understanding? I don't know if I could.
So the advantage of using a combination of TMS and EEG, that's your question?
Yeah.
Okay, so there are a lot of advantages using a combination of these two techniques, since you can at the same time modulate the brain activity, okay, in a non-invasive way. So you are modulating the ongoing electrical activity, and at the same time you are measuring the induced cortical activation with a really high temporal resolution. So and in some way you are you can also characterizing more or less the region where the TMS activity spread in the brain. So I'm not saying that when you find an effect in a certain electrodes, you can say that this effect comes from these electrodes, but for sure you can observe a pattern in different conditions.
Okay, but I was also, yeah, thank you very much. Yes, after this, I was thinking that is it possible that there are some artifacts in the measure of the EEG because of the stimulation of this magnetic field?
Yeah, a really huge artifact. But over the last 10 years, EEG, Javi, help me, EEG analysis method has improved a lot, and for example, now we can use independent component analysis, which allow us to clean the signal much better than 10 years ago. And then, so basically what you do is recording the EEG with a really high and in total would be 10 milliseconds. So you're losing just 10 milliseconds, and then with the multiple round of independent component analysis, you can clean your signal. And of course, you have to interpolate that. You can interpolate that time window. But if you're not interested in the first 10 milliseconds after your event, then you will have a clean signal also for registering TMS and EEG.
Yes.
Thank you.
Yes, it is the possibility to use some, as I tell to my students in the EEG course, that you can use some new techniques by applying algorithms to clean your signal when you are sure from where is coming from your artifact. No? So it is possible. So. And I'm not expert in TMS, but I remember that you, Chiara, and Fede were working in trying to clean the signal years ago. Yeah. And
there are more and more toolbox for this, so to better clean TMS artifact.
Yes, for sure. Thank you so much. Is there another question? If there are not more questions, I have the last question that is most, I don't know, maybe it's not really interesting, my question, but I want to know because I never asked to the patients, what is the experience, the emotional experience when they see something in the blind field?
I have extensively tested SL over this year, and she was really, really happy to report something coming from her blind field, because patient affected by visual field defects experience this visual hemifield, which is not, is a restricted visual hemifield, okay? So they know that something is missing, but they don't have any gray area like we can imagine maybe. So to perceive something is like our perception of the space behind us, okay? So I'm not perceiving that something that is behind me is missing. It's just not there. I know that there is something, but my visual field is restricted to the space in face. The space around me. So it was good for them to report something.
Yes, okay, thank you. There is another question from Dirección CINCCO.
Yes, I'm sorry, but this is very exciting, all this. Merleau-Ponty claimed that if you're in front of a cliff, and then you turn around and you don't see it, he claimed that the cliff itself is part of your perceptual field in a sort of large sense. I would perhaps say that hemianopic or some kind of lesion patients perhaps also integrate the missing part as part of their experience in a non-phenomenal way. But I mean, this is of course to be demonstrated empirically. But I think it's very interesting that the emotional part or the sensing of the missing part might also be part of the experience, not of the visual field, but of the experience as a whole. And then I wanted to ask whether you think that other kind of Phosphenes, like induced Phosphenes, when you press your eyeball, you also have experiences of phosphenes, and also speleologists that spend several days beneath the earth. They also claim to have spontaneous images, forming. So would you say that those kind of phosphenes that are produced by the retina pressure, by pressing the retina, or by spontaneous activity, are of the same kind of the TMS induced phosphenes?
There are different aspects of this same phenomenon, I would say. Of course, if you press your eyes, you experience phosphenes, but in this case, phosphene is generated by your retina, okay? But if you are, Javi, can you help me, speleologist? Is that correct? I can't remember the name.
Yes.
Okay. In that case, phosphine are generated. I would say that phosphine are generated by visual cortex, which is in light deprivation, and so it's like when you go to bed and you close your eyes, and it happens to me at least to close eyes and to perceive phosphenes, and they are more or less the same. So in that case, I would say they have a cortical origin. So is your cortex, is your visual cortex that is working and producing something, especially when you don't have light.
Exactly. So I was wondering whether they were phenomena of the same type, phenomenologically, the impression. It would be probably interesting to compare these different types of experience.
Do you mean comparing the two phenomena? So the same phenomenon with different origins?
Right.
Okay, that's interesting. We should try.
Yeah, you can try with. You have a lot of machines in your lab. You can try.
Different toys.
Yeah,
Yes.
It's about machines, but it's also about reporting, describing your experience, and for that, a phenomenological training also is important.
Yes, and also when we have new patients, for someone it's really easy to report phosphenes, but reporting phosphene is also something related to the personality. I mean, if you are a person particularly, I would say, I don't get the word. Particularly,
introverted, chilly.
Not liberal. The opposite of liberal. Sorry, in this way, I
a conservative person.
If you are a conservative person, probably you will say me, okay, I don't know what you are asking me to see. So what are you asking me? I can't see anything. It depends also on the kind of the personality you have.
Yes, I remember that you also have an experiment trying to compare conservative and liberal decisions for perceptual awareness. Mm-hmm. That's true, no?
Yes, so the response criterion is really important in this case. And that's why we always perform a training before performing phosphene experiments, so that they can really understand what they are required to report. And then the day after or the week after, we will perform the real experiment. But sometimes it's necessary to understand what a phosphene is. So despite it's described as a light sensation, if you ask participants, to report light sensation or flashes, they never see nothing because they expect something really bright, really impressive. But Phosphenes are really faint, really short, really brief percept. So also the expectation are really important reporting Phosphenes.
Yes, actually, Juan says it is interesting the link between personality and reportability. It has been shown that perception of stereograms and 3D popping figures are also function of personality.
Yeah, I don't know anything about this, but I would say that the two phenomena are related. And there is also a kind of new new field that say that this feature depends also on the size of your primary visual cortex or your visually responsive area. So probably measuring the extension of your visually responsive areas, you can also correlate with what you experience. And also attentional resources perhaps influence the reporting experience of Phosphene, for sure. But attentional resources do affect the reportability of this kind of percept.
Okay, Dr. Diana Plata say thank you so much for sharing your time and experience with us. Congratulations. And
Thanks.
as you read, also Juan say that attentional resources, but maybe also other cognitive processes can influence the reporting experience of Phosphenes. So they say thank you so much. And the last question is regarding the availability to share your experience with our students in terms that we can open collaborations with your lab and exchanging our students with you in your lab. It is possible?
Yes, it is. Email me.
Okay. So you are welcome here in the Centro de Investigación en Ciencias Cognitivas when you want to come and when the COVID possibilities will be better. And also, if you can receive our students interested in this field in your lab, it will be a good and excellent collaboration. So to finish our seminar, just I want to share the certificate.
Yes, thanks.
Just let me find it. Okay. Okay, this is a certificate for your Georgios presentations. So it say, I will read it in Spanish, okay? El Centro de Investigación en Ciencias Cognitivas de la Universidad Autónoma del Estado de Morelos otorga el presente reconocimiento, that means certificate, a la doctora Chiara Mazzi, Department of Neuroscience, Biomedicine and Movement Science, University of Verona in Italy, por su valiosa participación como ponente en el Seminario Transdisciplinar, impartiendo la conferencia Investigating Perceptual Awareness by Means of Phosphenes. Cuernavaca, Morelos, a 25 de octubre del 2020, por una humanidad culta, una universidad de excelencia, y firma el Dr. Juan Carlos González González, director del Centro de Investigación en Ciencias Cognitivas. So, thank you so much, Chiara, for your participation, and I hope we can share these scientific spaces in another time, in another moment. So,
thank you for your attention and for the invitation again. It was a pleasure for me.
Okay, thank you. Ok, muchas gracias a todos por su participación. Pues damos por terminada la sesión del seminario del día de hoy. Y pues nos vemos en los siguientes días y en el siguiente seminario. Creo que este es el del último año. Nos veríamos hasta el siguiente año en el siguiente seminario transdisciplinar. Muchas gracias. Bueno, thank you so much. Ti ringraziano e ti salutano tutti.
I'm reading now.
Abbiamo un commento che dice grazie per la charla, è stata molto interessante, saluti da Cile. Ma ti hanno sentito nel Cile?
Cos'è la charla, Javi?
Their talk.
Ok.
La conference. Okay. Thank you so much. So, see you the next, guys. Colleagues around the world, in Cuernavaca, in Italy. See you the next.
Gracias, doctor. Hasta luego. Gracias, doctora.
Gracias.
Bye. Thanks again. Bye.
Okay.
Gracias.